Well, we are going to be talking today about the lactose operon of E. coli. Now, the lac operon is under both positive and negative control. And we are focusing for this part just about the negative control, which means we're going to be focusing on the repressor molecule. So this is our E. coli chromosome. As you can see, it is a piece of circular DNA that is in helical form. And this is not to scale. So we have our lac operon here. And remember, what is an operon? An operon, of course, is some structural genes that are controlled by a promoter. A single promoter that is controlling several structural genes. Now, one of the reasons why we wanted you to visualize this as the whole chromosome is that a very important part that I want you to remember is that the repressor molecule is not part of what we consider the operon. The repressor molecule is found separate from everything else. The repressor molecule has its own little promoter, and it's made constitutively. That is, it's made all the time. Okay? So this is known as the LAC-I gene, right? And it's separate. So it's being made all the time. All right? This is, we've just put it here. That's an arbitrary thing. And so we want you to remember this. Again, not to scale. But just as a reminder that repressors and activators that are associated with operons are not actually part of this whole unit that we call the operon. Well, we're looking now at our setup for the operon, and again, we're talking about the lactose operon. We assume that you've already seen the first film about operons in general, and so that you're very familiar with all of this terminology. Now we're talking about the lactose operon. And so we have our promoter and the operator region, and we know that you're familiar with these terms and what these terms mean, and that you understand that the template strand is the strand that is going to be read by the RNA polymerase when it binds to the promoter. In the lactose operon, we have three structural genes: the LAC-Z gene, the LAC-Y gene, and the LAC-A gene. The product of the LAC-Z gene is a protein called beta-galactosidase. Beta-galactosidase is very important because let's take a moment and just remind ourselves, what is the lactoperon? What does it do? The lac operon is a very important operon when the sugar lactose is around. This is the sugar lactose, kind of, sort of. Lactose is the milk sugar. It's a disaccharide. And when lactose is around, lactose can be used as an energy source by the cell. It needs to be broken down and then used. Great idea. Okay. So remember, operons exist in the cell to put all of the genes under the control of a single promoter. We don't want the genes expressed if we're not going to use them. And that's what's great about an operon. Okay, so here we are. Three genes. What are these genes? What do the gene products do? So the first one, LAC-Z, the protein product is called beta-galactosidase. Beta-galactosidase is a protein that will take lactose and split it into two monosaccharides. Very important. LAC-Y is a permease. Right? And this is a protein that's going to help allow for the transport of lactose into the cell. Finally, LAC-A. Now, LAC-A is an acetyltransferase. And within the actual lac operon, its role is a little bit not well understood. Most acetyltransferase actually work by adding acetyl groups to sugars to have them exported outside of the cell if they are not being used. Its role in the lac operon, not so well understood. Okay, so we mostly concentrate on lac Z and lac Y. So these are our three structural genes that are part of the lactose operon. If no lactose is available, no lactose, then we don't need these genes. Why waste the energy? We don't need these gene products. Why waste the energy? Waste of energy, and if nothing else, E. coli, most bacteria in general, very efficient. We do not waste the energy to transcribe and translate this. All right? So under most circumstances, we have, remember, outside of the operon proper, we have the lac-I gene. And it codes for the effector molecule. And in this case, the effector molecule is that repressor. So that puts this operon under negative control. The repressor is active, right? So it will bind to our operator, and even if RNA polymerase is around, we are not going to get transcription of this operon. It is under negative control. No lactose around, no transcription, everybody's happy. Now the lactose operon is an example of an operon that is under negative control, that is an inducible operon. All right, so lactose, actually the form of lactose that is known as allolactose, is what we call that modulator. Remember we talked about a modulator? And in this case, because the operon is inducible, then this is known as an inducer. Now the specific form of lactose that is the inducer is actually called allolactose. So what does this mean? Lactose, because it is the inducer, lactose is available in the systems. Lactose can bind to the repressor. When lactose or allolactose binds to the repressor, it makes a conformational change on the repressor molecule. And when that happens, the repressor molecule no longer binds to the operator. When that happens, now the operator area, that sequence is unbound, and now we can get transcription of our structural genes. Because we can get transcription of our structural genes, the three gene products necessary for the breakdown of lactose, beta-galactosidase, permease, acetyltransferase, all three of these gene products these three will be transcribed in one single messenger RNA. That's a polycistronic message, which will then be translated, and we will make our three gene products. A question a lot of students ask is that the product of the lac-Y gene is the permease, and how can lactose actually get into the cell if the lactose operon is shut down? And the answer is that the repression is not complete. And in many cases, the lactose operon, because it is not completely shut down, there's a small amount of beta-galactosidase, permease, and transacetylase in the cell. So that if lactose is present, a small amount of lactose will be converted into allolactose. And that allolactose can bind to the repressor molecule. And so the whole process of that repression being the allolactose binding to the repressor molecule and beginning this process of induction, that will happen. One of the fun things about the lactose operon is that it's under negative control, as we've just explained. Once we have lactose in the system, we bind to the repressor molecule here, and we've opened it up. here's the really interesting thing about it. We've done this. Lactose is bound to the repressor molecule. We already talked about the fact that oftentimes when a repressor is in charge of an operon and we have repression, we've talked about the fact that we have weak promoters and strong promoters. The lactose repressor is actually a fairly weak promoter. Who knew? Who knew? So it turns out that even if the repressor is lifted, we sometimes get pretty poor transcription. It's very sad indeed. And so it turns out that the lactose operon is not only under negative control, it's also under positive control. And one of the reasons it's under positive control is that even if lactose is in the system, many times you do not want to be transcribing this particular operon. The only time you want to be transcribing the lactose operon is if lactose is the only carbon source available. If there is another, better carbon source available, and the carbon source that we're talking about, of course, is glucose, then we don't want this, because lactose, good, glucose, better. And so this is an example of a type of control that is both positive and negative control. And this is known as catabolite repression. So we're going to talk about how this is under both positive and negative control. And what controls this is the levels of glucose in the system. E. coli would rather have glucose than anything else. And let's face it, wouldn't you too? As we talk about activation in the lac operon, one of the things that we want to remember is to talk a little bit about glucose, because glucose plays a role in this. So here's our glucose molecule, and this is something that we want to remember because glucose plays a role in ATP. And so the levels of glucose determine something about the levels of ATP. If our levels of glucose are high, then the levels of ATP are going to be high. And as the levels of glucose drop off, as the cell doesn't have glucose, then instead of ATP, ATP is going to be broken down. And then in the presence of an enzyme called adenyl cyclase, we are going to make cyclic AMP. And that's what this is here to remind us of, the cyclic nature of this. Okay, we're going to make cyclic AMP. And it is cyclic AMP that is going to be very important in the lactose operon. All right? So this is a very important, and this is what we want you to remember. When glucose levels are very high, we do not want to be using anything else except glucose. That's what the cell wants to use. But as glucose levels are depleted, then cyclic AMP levels become very high. And so cyclic AMP levels are going to be high in the cell, so there's going to be a lot of cyclic AMP around. And that's what we're going to be talking about. Okay, so here we are with our lactose operon, and we have a situation where glucose is available and so is lactose. So lactose binds to our repressor molecule, and it is lifted. But glucose is available in the cell, and so we have an activator protein. But the activator protein is inactive right now. It's not able to bind. And because it's not able to bind right now, we're not getting really good transcription. All right, the RNA polymerase, remember this is a weak promoter, the RNA polymerase isn't really binding very well, so we're not getting efficient transcription. And that's because there's glucose available. And because there's glucose available, we're making ATP, and we're a happy camper. But glucose is slowly being dissipated in the cell. And we finally reach the point now where there isn't much glucose available, and Now what we're starting to build up is cyclic AMP. And over a period of time, oh dear, that ATP is going away, and now what we're making is cyclic AMP. And as the cyclic AMP builds up, it will bind to our activator molecule. In the lactose system, this activator molecule is called the CAP protein. CAP stands for Catabolite Activator Protein. Now, the cyclic AMP is going to bind to it, and when it binds to it, it makes a conformational change which allows it to bind to the DNA. And when it binds to the DNA, it helps that RNA polymerase bind. And when it helps the RNA polymerase bind, we now get really efficient transcription and then translation of our lactose operon, which is what we want because remember, we have lactose in the system. Oops, everything fell apart, but that's okay. We have lactose in the system. And now, because there isn't any glucose available for energy anymore, we want to be able to use our lactose that we have in the system for energy purposes. So this is how the lactose operon is under both positive and negative control at the same time.